Hello, and thank you for participating in the poster series entitled Quantitative Live Cell Imaging Assays for Immunotherapy. My name is Laura Skurlos, and I'm an application scientist at Sartorius. Today, I'll be presenting a poster entitled Dynamic Live Cell Visualization and Quantification of Cell Cycle and Pharmacological Response using a Genetically Encoded Ubiquitination-Based Sensor. All of the data presented in this poster involve the use of the Incucyte system for continuous live cell analysis. Using this instrument, researchers may conduct real-time measurements from inside their own incubator. Along with the instrument, the Incucyte software offers fast, flexible, and powerful tools for analysis. The Incucyte reagents and consumables are also offered at Sartorius, and today we'll be describing a genetically encoded cell cycle sensor using a Lentivirus vector. The Incucyte green-red cell cycle Lentivirus reagent encodes a fluorescence ubiquitination-based cell cycle indicator, otherwise known as FUCCI. This multi-gene expression system allows us to visualize cells as they progress through the cell cycle. This schematic illustrates how the fluorescent protein expression changes over the course of the cell cycle. In G1, the cells express the red protein. During S, G2, and M, the green protein is expressed. And in the overlap of G1 and S, the cells will appear as yellow. During the stage where the cell is cycling from M into G1, there is a short period of colorlessness. The figures here show images of HeLa cells transduced with the cell cycle red-green lentivirus reagent. You can watch at time 0 as the cell starts in G1 as a bright red, and at 2 hours and 4 hours you can see the cell cycling through G1S, turning a yellowish orange, and then between 7-9 hours you can see the cell is cycling into SG2M, and you can see the distinct morphological change at 9 hours, and then the cells will split back into G1, where there are 2 cells now. The next panel of experiments highlights the use of cell-by-cell software with the cell cycle indicator. In the first panel, Incucyte cell-by-cell acquisition shows how it enables accurate masking of individual cells. The yellow segmentation mask is shown here. Using this segmentation mask, quantification and classification of the cell population of each phase is shown in using the scatter plot. On the x-axis is the red mean intensity, on the y-axis is the green mean intensity, and each dot represents a cell fluorescing. Where the cells are expressing both red and green, those appear in the yellow population, representing the G1S overlap. The mask overlays using cell-by-cell software illustrate an even distribution of cells. In the last data plot shown here, you can observe how the population of cells changes over the course of time. Using a 24-hour thymidine synchronization protocol, HeLa cells were arrested in the S-G2M phase of the cell cycle. In the plot, you can see the population is held in the green phase in the early time section of this plot. Upon release of the thymidine block at just past 24 hours, the cell cycle progresses through M, G1, and then back through SG2M. Using this type of analyses, you can determine the cell cycle, in this case about 17 hours, which is the peak-to-peak duration. As with our entire suite of live cell imaging reagents, the cell cycle reagent is non-perturbing. Shown here are images of parental MDA MB231 cells and stable cell lines expressing the Incucyte red-green cell cycle lentivirus reagent. By looking at the images, you can see no distinct change in morphology from the genetically encoded cell cycle reagent. Further, the growth kinetics of the parental strain and the genetically modified strain are similar. The data for 2 cell lines, an adherent and a suspension cell line, are shown above. Proof of concept data using 2 well-characterized cell cycle disruptors is presented using microplate plot format. Cisplatin is an intra-strand cross-linker which disrupts DNA replication and is known to hold cells in the SG2M or green phase of the cell cycle. 5-fluorouracil, or 5-FU, is a thymidylate synthase inhibitor and known to stall cells in G1 or red. Microplate plots were generated from images analyzed with cell-by-cell analysis software. The plate data shows the percentage of cells that are red, green, or yellow. For each plate, cells in columns 1 through 6 are treated with cisplatin, and cells in columns 7 through 12 are treated with the 5-FU compound. Vehicle-treated cells are in row H. These microplate views illustrate the concentration and time-dependent effects on cell cycle over 30 hours. Specifically, the 5-FU data illustrates the expected increase in red classified cells, whereas the cisplatin illustrates an increase in green. Using the HT1080 fibrosarcoma cell cycle distribution data, concentration response curves are shown for populations in G1, SG2M, or the G1S overlap. The concentration response curves for cisplatin at 24 hours post-treatment show a decrease in the G1 phase and increase in the SG2M phase. 5-fluorouracil treatment is opposite, as there was an increase in cells in G1 and a decrease in the SG2M phase of the cell cycle. Visualizing the HT1080 cells with phase and fluorescent imaging, you can see that after 24 hours of drug treatment, cisplatin holds the cells in the SG2M, or the green, whereas 5-FU is holding the cells in the red G1 phase of the cell cycle. Segmentation analysis using cell-by-cell software is visible here, using yellow masking. The genetically encoded cell cycle sensor was also used to analyze selective drug effects in breast cancer cells. Here we tested 3 chemotherapeutic compounds on 2 cell lines, a triple-negative breast cancer cell line, MDA-MB231, and a receptor-positive cell line, T47D. The concentration response curves show the percentage of populations noted by the color of the line, so for example, red would be a G1 population, in each cell population treated with the different chemotherapeutics. Tamoxifen is a known estrogen receptor therapeutic. The ER-positive cell line, T47D, demonstrates a disruption in the cell cycle and a hold in G1, whereas no change in the cell cycle is observed with the MDA-MB231s. CB839 is a glutamase GLS1 inhibitor currently in clinical trials to treat triple-negative breast cancer. CB839 induces an anti-proliferative effect in MB231 cells by arresting in G1, while the cell cycle in T47D cells is not disrupted. Carboplatin is a nonspecific inhibitor of DNA synthesis, interrupting DNA replication. Both breast cancer cell lines are held in the SG2M phase of the cell cycle when treated with this chemotherapeutic. Taken together, these data show the Incucyte cell cycle indicator can distinguish selective chemotherapeutic effects. The cell cycle fluorescence indicator was used to look at several aspects of THP1 differentiation, including proliferation, morphology, and function. Shown are THP1 monocytes stably transduced with the cell cycle lentivirus reagent. Cells are treated with either vehicle or PMA. Using cell-by-cell software, proliferation and cell cycle were determined. Plotting cell count over time shows that treatment with PMA induces a lack of proliferation. The disruption in cell cycle after PMA treatment is also shown. By plotting the percentage of population in either the green or the red phase of the cell cycle, a clear increase in the percentage of red cells, or the G1, and a clear decrease in the green cells is shown with the PMA treatment. The morphological change induced by PMA in THP1 monocytes can be visualized in the image as well as the cell area plot. With regard to functional differences of THP1 differentiation, phagocytic potential was measured by efferocytosis. Here, URCAT cells labeled with Incucyte Frodo red labeling kit show a higher degree of apoptosis with the PMA-treated THP1 cells as compared with the vehicle-treated. A final application of the Incucyte cell cycle lentivirus reagent is three-dimensional imaging of dynamics in a tumor multi-Spheroid model. A human lung carcinoma cell line, A549s, were seeded on top of matrigel and allowed to form multi-Spheroids for 3 days prior to treatment with compounds. Shown here are the multi-Spheroids treated with either vehicle, 5-FU, or carboplatin, with and without the phase. Multi-Spheroids treated with a vehicle show the cell groups arrested in a diverse population of cell cycle, green, red, and yellow. Cells treated with 5-FU are arrested in the S phase. Most cells are shown red. And when the multi-Spheroids are treated with carboplatin, the majority of cells are held in the green S-G2M phase. So wrapping up, I hope you can see how the Incucyte green-red lentivirus reagent is an effective and relevant tool for studying cell cycle dynamics in real time. The cell cycle reagent yields stable, non-perturbing expression of green or red fluorescent proteins, and using the Incucyte cell-by-cell software module, pharmacological disruptions in cell cycle can be quantified. Thank you so much for your time and participation in this poster series.